Okay, now we should go through an example report. So I'm going to jump back over to here. This is a report from a field where we did a trial, and I'm going to actually go through it kind of in chronological order. It's like if we were working together. So let's start with the prescription. So this is a fungicide trial. This is on a large Midwestern farm, and they grow mostly corn and soy. And they said, take 4 of our 80-acre cornfields and measure as precisely as you can whether this new fungicide pays off in our area. And they're large enough that they sort of negotiate on all their inputs, and so the price on their inputs, and so they said that we're going to use this for a pricing negotiation next year. So you see, we fit in 70 replicates. The light green is no fungicide. The dark green is with the fungicide. And they have nice equipment, and so we could basically make these exactly 2 combine passes wide, and they're fairly small. These are roughly maybe 3/8 of an acre in size. So that is the prescription, picture of the trial, and then there's a variable, the shape file of the prescription that they load in the rate controller. And so they load that in the rate controller, they load the fungicide, they get to the field, and they apply it. So you can see here that they did a really nice job when they applied it. The one, you could call it a mistake, but it's not a big deal, is where they entered the field, the pressure was a little high, and then it quickly got to the right rate, and then just, the valves turned it off and on as it went through the field, and they did a great job. We like to get the as applied data. We don't have to have it. We can analyze the trial without. We'll just assume that the trial was done perfectly. But when we can, we like to look at it, because sometimes we find, maybe we have to throw out 3 or 4 of the replicates. There were mistakes made. And so that's why we like to see it. Finally, at the end of the season, we get the yield data back. And so a lot of the growers that we work with and customers we work with will say, or the ag input companies will say we don't want to do small plot trials for our early stage research after it comes out of the lab or out of the greenhouse. But then right before we are going commercial, or after we've gone commercial, we like to do tests and product demos. And normally what we'll do is we'll take half the field and put the fungicide on, and half the field and don't put the fungicide. But you can see really clearly in this example that the difference between the west half of the field and the east half of the field is bigger than the difference between having the fungicide and not. And so that's why we are proponents of lots of small plots in a replicated design, that it lets you tease out more of the signal from the noise. Okay. So for each of these pairs of plots, what we do, we take the yield from the plot with the fungicide, we subtract the yield from the plot without the fungicide to get a yield difference. Those yield differences are what we're analyzing. So kind of what you can think about here is, it's like we are doing a randomized control trial, but you can imagine doing a medical trial where you test a drug and you would be testing on pairs of twins. That's what we're trying to do here. We're trying to, that's the same sort of math that we're doing here. We're basically doing we're looking at the differences between 2 pairs of plots. Okay. So we take those 70 yield differences, and that's the data that we're going to analyze. And that goes into this histogram. And so this is the most important graph in the report. This is those 70 yield differences shown in a histogram. And so you can see here that 0 is in the middle. And so 3/4 of those yield differences are positive, and 1/4 are negative, or around 0. We like to have lots of replicates. So not only does it tell you where the center is really precisely, but it also tells you how broad that distribution is, so that you can tell how confident you can be in that result. We do the same with the profit, and we summarize that in a table like this. So you can see here, if we focus on the yield column first, we have the yield with the fungicide, the yield without, and the observed difference. And I'd say lots of software will get you those numbers. But what's unique about our offering is that we also produce a least significant difference, an error bar. So if your observed difference, as in this case, is larger than your LSD, then you can be reasonably confident that this is not due to randomness. This is very likely reproducible, especially when the difference is as big as this, a factor of 4 or 5, then you can be really confident that this result is believable. And actually, in this case, we did it on 4 trials, and they all sort of had a similar kind of, a big observed difference compared to the LSD. In contrast, if you do on your trial and your observed difference is smaller than your LSD, that's where you have to say, there's a good chance that this trial won't reproduce. And so you have to take that kind of data with a grain of salt. So anyway, we do that analysis of the yield with the profit, and then we try and get just really clear and concrete that, in this case, Mycosine Pro produced a statistically different profit and yield result from not applying it. Last thing on the report, we also pull in the weather from, so the precipitation and the growing degree units from a nearby government weather station. We feel like this is part of providing context about what happened in the field that year. And then we also create this agronomic and experimental summary. We like to have this so that, in 5 years you come back and you're trying to figure out, try and remember, like, what was the variety that we used on that trial? What was the planting date? What was the exact tank mix? We like to get that all in one place so that you have it and it can reference it. And, in the here and now, we're really focused on helping you understand which of a couple of management practices paid off on a specific field on a specific year with that weather. But we're trying to build a really nice curated data set so that you will be able to figure out within your growing operation or within your sales area, where the products are, where you should place products for them to have maximum benefit and where you shouldn't. And so we are trying to basically help companies of all those types, researchers, growers, crop consultants, make those data sets they need in order to be able to make better predictions about where to place products. Okay, that's everything on the grower report. I guess, just that's the whole report. The whole presentation. I'm happy to jump into questions and answer in here a second, but I want to say the last thing. If you want to discuss your needs in more detail, you can go to this link and we'll share that in an email with you. You can click on that and so you can schedule a little meeting with us to talk about how we can help you.